Ena mana, ena reo, e rau rangatira mā, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora koutou, I'm Sarah Young. I'm the Dean of Science here at Waipapa Taumata Row, the University of Auckland. It's really an honour to welcome you all here today and to introduce Professor SallyAnn Harbison, a scientist whose work in forensic biology has helped shape the very way that sciences serve justice. After completing her BSc and PhD in biochemistry at the University of Liverpool, SallyAnn moved to Aotearoa, New Zealand in 1985, the very same year that Alec Jeffreys' discovery of DNA fingerprinting revolutionised forensic science. It was a moment of scientific transformation, and SallyAnn was poised to be part of it. Though she began her career as a plant RNA virologist, a postdoctoral position in Richard Gardner's cellular and molecular biology group at the University of Auckland sparked a shift to forensic science. Initially, both a forensic biologist and a crime scene examiner, SallyAnn is today one of the country's leading figures in forensic genetics, specialising in body fluid identification, DNA and RNA analysis, and interpreting complex biological evidence. SallyAnn is a senior science leader in the Forensic Biology Group at the New Zealand Institute for Public Health and Forensic Science, formerly ESR, and she's also the director of the Forensic Science program here at the University of Auckland, having started as an honorary lecturer in 1996, supervising more than 70 student researchers along the way, and an absolutely outstanding example of great partnership between the two institutions. Her contributions to forensic science have been recognised nationally and internationally. In 2021, she was made a member of the New Zealand Order of Merit and a Fellow of the Royal Society, Te Aparangi. In tonight's lecture, SallyAnn will offer us a personal and professional reflection on a career spent at the intersection of research, professional practice and justice. It's a story of relationships, of resilience, of innovation and of teamwork. Thank you all for being here today, and please welcome Professor SallyAnn Harbison to the floor.
Okay, thank you very much, Sarah, for your kind words. More or less done my talk for me. Marvellous. And also Anna, I know it was a big ask, thank you. Anna is in the office next to me at ESR, and I did ask if she would be able to contribute to this today. Good job, well done. Okay, so I hope that you are all enjoying yourselves as much as I am. I will not keep you long. My personal journey covers, as Sarah's already said, crime scene to courtroom, and the innovation of technology that underpins the forensic science services that we provide. And today I'm just going to give you a snapshot of what we do and what I've been privileged to be part of. So, some disclaimers. This is a forensic talk, and it is a forensic biology talk in particular. So I have tried very hard not to upset or offend anybody, and I really hope that I have managed to do that. It's not exactly family friendly, but it's close to it. There may be tears. Don't worry about that. My ESL colleagues know that well. Just roll with it. It'll be fine. And I will have omitted things. I have had to omit things. I couldn't include everything, and I probably omitted some people. And if you're here and I've done that, I'm really sorry in advance. I probably, Sarah, I'm sorry. I will probably say ESL a few times when really I should say PHF science. It's not quite as easy. Really. So I apologise for that too. And lastly, I must apologise for some of the photographs are old and have had to be rephotographed and may not be as sharp and clear on a big screen as they are on a laptop. So let's go. Okay, first of all, I wanted to thank all of the students that I have supervised directly in the forensic science program and some other universities as well. Master's students on the left, PhD students on the right. The ones in green came to ESR directly from their studies. Some of them have moved on since. The ones in blue are with me now, and the ones in purple went straight back to their home countries to start doing forensic science practice overseas. And my PhD students are a wonderful group, and I couldn't have done any of this without them. And my dream team is here, bar one, I believe. Bethany, Becca, Alex, and Ryan. I don't think you're all here. I don't think Ryan's here. Unfortunately, we're in the school holidays, so bad timing. But this is a picture of us at Bethany's graduation just last year. Okay. I also want to thank, obviously, all my colleagues at PHF Science, who I've worked with, obviously, for a long time now. When I started, it was DSR Chemistry, and then we had an interim IEHFS. Now we're back to DSR, now we're PHF Science. 40 years, 4 names, I guess that's about right. But I really want to thank a couple of people in particular, Sally Coulson, who's here, and John Buckleton, who have been teaching on the programme with me for most of that time. Gordon Muskelly, front here, and Douglas Elliott, who's not here, for their leadership of the programme, and Gordon in particular for taking a chance and appointing me to be the director of the programme when he needed to. I'd also like to thank Margaret Lawton, who was my previous general manager some years ago, for taking on a heavily pregnant woman, seriously heavily pregnant woman, and in the first place, Keith Bedford, general manager in between, who steered me down the science leadership route away from management, which was probably a really good idea, although I didn't think so at the time. So many of you here have provided me with images to use, which I have. I've given credit to all of them, I think, in the right way. But I would like also to thank, in particular, Jenny Ralston from PHF Science, who is one of our comms people who does all our posters, all our brochures, everything. And she is responsible for some of these images, especially the one on my opening slide. So, also a massive shout out, doing the thanks up front, right, Kenzie, to my international colleagues and friends across the globe. It's been a privilege to work with all of them and get to know them so well. I particularly want to thank my friends at King's College London in the middle, Jack Valentine on the left from Florida, Peter Gill from Oslo, who else do I have here? Oh, and the ISFG, which is our International Society for Forensic Genetics, who have provided me over the years with many opportunities to do really good research, have really good collaborations, and make some really good friends. I also wanted to point out that I'm also the quality manager for my team at ESR, and so I have done audits all over the world, in North America, Canada, and Asia, Australia, and New Zealand. And I wanted to do a bit of a shout out for the auditors that I've worked with and the auditees, because their collegiality and openness to change is amazing and really demonstrates the quality culture that's needed in a forensic science organisation, and that applies worldwide. It's necessary, and it gives you more confidence in what we do. And a number of you here are also auditors that I know. So I know there's lots of people here from chemistry, and that means I know that there's lots of people here who have absolutely no idea what forensic biology is all about. So I've got this one slide which will give you some context. So you can kind of read it, really. But what we do is we collect samples and identify samples from all biological samples from a wide range of cases and things, from people, from crime scenes, etc. And we identify the biological source of whatever that material is. Mostly we use chemical tests for that, but sometimes we use messenger RNA, which I'll come back to later. Then we make DNA profiles from the crime scene samples and from people. Those DNA profiles can be either done with something called capillary electrophoresis, which you'll see in a moment, but also now more and more with DNA sequencing as well. And we compare those either to people directly or to DNA databases, including our national DNA database, which I will show you something more about in a moment. Then we do some stats. That's a very brief. Then we do some stats. And then we interpret the data and turn up in court and give our evidence in court. And that's a picture of me in one of the Bain trials. You can find that in the Herald if you want to go and have a look. But all of this is underpinned by quality management and research and development, and my role particularly has been at the intersection of the research and development, the quality management, and the casework. So, now we're going to talk about my unexpected journey. Some of you know I'm a runner, probably most of you. Occasional marathon runner, it turns out. And most of my research career from my PhD onwards has been pretty much exactly 42.2. So I thought this was a useful way to describe what I've done, and it turns out that the phases of my career fit quite neatly into this time frame as well. And you all know, if you are a marathon runner, I'm really sorry, I often ask questions and I'm really trying not to. So I'm not going to ask if any of you are marathon runners. But if you are, you all know, you cannot finish if you did not start. You can't start if you didn't train. You can't train if you didn't plan and prepare. It's just one of those things. And this is me at various stages of the journey, which from Auckland last year, which you will see in some of the slides. And so my unexpected journey begins. Okay, let's cut the tissues. All right, so one of my areas of expertise is forensic genetic genealogy, and family history is also a hobby. So I was born and raised in Oxford in the UK, and I know some of you here at least have studied there, if not lived and worked there, so you will recognise some of the things that I say in some of the next slides, or you might even recognise them from here. So on the left we have my paternal family, my great-grandmother, Elizabeth, and next to her is my granny with me, and then the baby is my dad with his dad. That is the only photograph I have of him. My granny was not your average granny. I suspect nobody's ever are really, but she was a single mum. She was married at least twice. She was a troop entertainer in World War I. She tried to run off and join both the circus and the IRA, or became the IRA. Before she was 20, she was a bit disruptive and a bit provocative, and she took no prisoners, and she was a tiny bit high maintenance, which some of you are going, got that. She gave me the gift of my dad, and my dad is this beautiful gentleman here, and Dad is insanely clever. He didn't go to university because instead he joined the Palestinian police force, British Palestine police force in 1947, which nearly killed him, literally nearly killed him, and shaped everything he did from then on. But he was on professional staff at New College in Oxford. That's a picture of New College with my son Ruben, who's here somewhere, and my daughter. So I spent my formative years growing up in and around the college, in the gardens, in the buildings, everywhere. I went to balls, I went to graduation ceremonies, I did a whole heap of stuff with Dad that many people don't have the privilege to do. And that is why when I see you come into SCS with your kids in the holidays, I think, yes, they're getting what I got. It was a fabulous experience, and I was really shaped by it. But Dad was into everything. He was so clever. He was into arts, he was into books, he was into music, he was into theatre, he was into maths and science and gardening and everything. I can't think of anything he didn't really like. He passed that all on to me, because I was the oldest, probably gathered that by now. And the only thing he didn't pass on was rugby, because I picked up football. And he wasn't very happy about that. So on the right, you're right, is my mum's family. So the three ladies are my great-great-grandmother, my great-grandmother, and my grandmother, who's the younger one of the three. And then my great-great-granddad and my great-granddad and their dogs, and my grandpa at the bottom. And then me and my mum, who's been to St Giles's Fair. No, I said I wasn't going to say that. So this is St Giles's Fair in Oxford, and if you've ever lived in Oxford, you will know St Giles's Fair. It's on every year, and that's a picture of me and Mum at St Giles's Fair. So Mum's family were completely different. They were from the country, not the town, and that's where my Hobbit comes from. For those of you who wondered where did the Hobbit come from, that's where it comes from. So before I hit primary school, at 3, I could read, I could write, I could do algebra and basic maths, and I was playing the piano. But during primary school, a few things happened that really shaped my life and influenced my choices. This is the beginning of the story, I guess you could say. I'm a child of the moon landings. Some of other of you in the room maybe too. and I really wanted to be an astronaut. I always wanted to be an astronaut. I still want to be an astronaut, and I haven't given up. So that was one of them. England won the World Cup, a little bit exciting for if you're English, and I became a member of the Liverpool Football Club family. And I have my badge right here. If you're part of that family, you will know you never walk alone, and there is no I in team. And we've taken some pretty hard knocks over the years, most recently last week, and we've learned to be resilient. And I credit a lot of my resilience, some of you think I don't have any, but I do, with belonging to this particular family. There were some of you in the room I know. I have met so many people through this family. It's amazing. It doesn't matter where you go in the world. And then I have to mention that my brother arrived, and that's me and my little brother. Not so little anymore. And then I went to grammar school, and of course I was a girly swat. I don't know if that's an appropriate term anymore, but I was. And I had some inspirational teachers, Mr. Heddy in maths, Mr. Sweet in chemistry. But I could have done anything. I could have done arts and music, or I could have done science. I basically had the option to do everything. I added the guitar and the trombone to the piano. I joined my brother in a brass band. I was a Queen's Guide, this one. I was a dancer in the gang show, the Oxford gang show, and I was a deputy head girl. I also found Tolkien, and this has probably been one of the biggest influences of, maybe other than the football. Not Tolkien, Lord of the Rings necessarily, although I love Lord of the Rings, but Tolkien generally. He studies philology, which is words from ancient texts. I love words. I love meaning. My ESL colleagues, I read their work and go, no, now we know where that comes from. I'm very particular about things. And I have a massive collection, and if you want to borrow it, you're very welcome, including, you can't have my Lego. You only have my Lego. But you can see in that picture there. So what do you do when you get to the end of high school? What do you do? And my students ask me this all the time, what do I do next? What do I do next? So I did the only thing I could have done really, I tried to capture as many of my interests in one go, and that's how I rocked up at the biochemistry department of the University of Liverpool at the end of the 70s. And Peter Sutcliffe, the Yorkshire Ripper, was active in the north of England when I arrived. That was my first exposure really to forensic science, I would say. We were given self-defense lessons in the Hall of Residence on the day we turned up, basically. But Liverpool at the end of the 70s was humming. The music scene was insane, and I took full advantage of that. I still have my vinyl at home. Liverpool were riding a wave of European and domestic success, and I was on the cop, if you know what that means, for every game for 4 years. Whilst I was an undergraduate, some pretty major shifts kind of happened. First of all, the department renamed nucleic acid biochemistry to molecular biology. So that happened while I was there. But also, I remember exactly where I was when I found out that mitochondrial DNA was maternally inherited. And I put the reference up there for Tegan and Beth, if they want to read it, if they haven't already. I hope that they have. Another thing that happened was I set my hair on fire in the genetics lab. The demonstrator for which was Peter Gill. And some of you will automatically know that name, but you will hear it again several times. And my honours project was on proteins and RNA. Protein synthesis, proteomics, and RNA. And that becomes kind of important as we go forward. So, this is kind of the end of the training stage now. We're now getting into the kind of race itself. But what did I learn at this point? There is no I in team, and you don't walk alone. What you do with words is really important in your work life and at home. If you're curious about stuff, and the more you know, the more options you will have. You don't know who you're going to meet, meet a girl who might become important later on. RNA doesn't have to be worked on in a cold room, but it does help. And just like a lamp assay, and if you don't know what that means, it doesn't matter. Life can come full circle. Okay, so first 5K. This is when you get warmed up and your rhythm gets going. I started off in Liverpool for my PhD, but it was a case studentship, which means it was an industry partnership. And after a year, I moved with my supervisor from Liverpool to the John Innes Institute. If you don't know the John Innes Institute, it's one of the preeminent plant research institutes in the world. It was right next door to the Food Research Institute in Norwich, which is also similarly positioned for food. And happily for me, also the Sainsbury Arts Centre, which is a gallery sponsored by Sainsbury's supermarket on the East Anglian campus. And my work was on carnation model virus. Is that beautiful bright green thing there? I never saw it look like that, but anyway, that's what carnation model virus looks like. And if you think it looks like COVID, yes it does. It's an RNA virus just the same. I was supervised by Michael Wilson and Jeff Davis, and I did all sorts of things there, but the kind of cool thing that I did there was direct RNA sequencing. And that's a page on my thesis, which is probably a bit blurry because I had to try and photograph it. We didn't have digital anything in those days. And the top left is called wandering spot analysis, and it is basically a 2 meter high thin layer chromatography analysis of RNA for RNA to sequence it. So those of you who do RNA sequencing different ways now can maybe think that that was probably so I did RNA and protein work again. I got to work with amazing plant virologists from across the world, including my future president Brett, who was a Kiwi. I was trying to make carnations resistant to the virus, in case that's what you think. I wonder what I was trying to do there. And yeah, so basically that's what I did. And that led me to the Department of Cellular and Molecular Biology, as it was then, and as Sarah's already said, with Professor Richard Gardner. And there I did yet again a university industry partnership. And I worked on a different virus, now one more kind of interesting to New Zealanders, being clover for agriculture, pasta, I should say. And I was there for 2.5 years. And I don't think anybody is here from that picture. I was kind of hoping some of them would be. Joe Petrel was probably the one that some of you might know. But towards the front is Douglas Elliott and Dave Saul. So that picture is from about 1986, or thereabouts. During my time here, PCR was invented, which is quite important for forensic science. And as Sarah's already mentioned, the first DNA papers were published. And I learned to read and memorize sequence in my head, because you had to read it off a jar and write it down at the same time, and I could memorize. I can still do that. I can memorize things like 40, 50, 60 base pairs long if I have to. Again, the first forensic DNA published. Papers were published, but postdocs don't last forever, and something has to start anew. So, this is my start of my forensic career, and if you haven't noticed, there's like a little bar across the bottom which will show you where we are in the journey. So we're at 5 to 15, right? So we're now warmed up, and we're getting ready to hit our stride. So, my first crime scene examination was with Lisa Amelia in the Henderson Police Station car park. She possibly doesn't remember that. My first homicide scene was in Whakamaru, down towards Lake Taupo with Robert Jingles, who some people here will know. I had my first baby, who's in the front, and my second, who's not in the front, but she's on the slide. I have my first deceased person, which was with Kevin Walsh, who was here too, somewhere I saw over here, which was in Waihi. That was my first dead body, by the way, Kevin, in case you didn't know. My first court case was across the road in the High Court in Auckland, in courtroom number 1, which is the most beautiful room I have ever been in, and I've been in some beautiful rooms. I also did my last crime scene examination, which was man versus train in the Otago railway yards, which was very pleasant. And in between the two babies, email and computers arrived. So that's a picture of me in the office I shared with Douglas, which is why it's so completely chaotic. We had no, yeah, they see, they know. We had no computers, there was no computer on my desk, there was no screen, there was nothing. It was all typed. So, in 1985, Alec Jeffries and co-workers at produce the first DNA profiling papers. And one of them you may be able to read was her first author was the very same Peter Gill. Led by the most amazing woman, Margaret Lawton, DSR Chemistry, including Sue and Lisa, who are here tonight, and possibly Kevin too, we validated DNA testing using the same fingerprint method that Alan Jeffries had developed. Some travelled overseas to learn in the UK, but Douglas Elliott and I were recruited in from the university. Other experts were brought in to help, so not only Peter and others, but critically Bruce Weir, who is also here at the back, who today spent his morning training our new generation of scientists in identical by descent and kinship and other forensic-related statistical methods. Bruce, like Peter, has been a lifelong friend to PHF Science, formerly ISAR, formerly DSAR, and also to me. So in 1990, which was the same year as the Aramoana massacre for those who are old enough to remember it, Margaret presented the first DNA evidence in a court case in New Zealand. Now, I'm not going to dwell on this slide too much because we don't need to worry about it too much, but this was the changing technology really from the time I started, which is old-fashioned blood grouping, ABO and other things, through the multi-locus probes that Alec Jeffries was using to something called single locus probes, which is a slightly simplified version of the same thing, and then on to what we are currently doing really, which was short tandem repeats and PCR. And I'm going to talk about that on the next slide. So a couple of things for the chemists to know. Short tandem repeats are little 4-base pair repeats, usually, not always, that repeat at different places on the genome, and different people have different numbers of repeats. And you get some of your DNA from mum, half from dad. You put them together, you get a DNA profile effectively. The slide before you were looking at gel electrophoresis. DNA profile that we've got up here on the slide is done with capillary electrophoresis and fluorescent dyes and computers. Doesn't quite look quite the same, but the output is similar. You either have 2 types that are locus or 1. 2 if you got one different one from mum and dad, and 1 if you got the same from mum and dad. And you should be able to see that some of those, that's 1 profile, some of them are 2, some of them are 1. But the really cool thing about this version of an STR profile is that it can be digitized. And that is what led to the creation of the DNA databases that we all know and love so well. Now, the biggest change from what we were before to where we are now, we didn't need a microgram of DNA anymore. Now we're in nanogram, sub-nanogram levels of DNA. And that was really the biggest change that was enabled by this type of technology. So I don't have very many case examples in here, but I do have this one. It was the STR technology that I developed in the early 90s that led to some of the work that was done in this case. And I put this case in because this has been a defining case in my career. It hasn't quite finished, it's not quite finished. I went to the crime scene in 92, so over 30 years ago, examined all the items. I did the blood grouping, because that's what we were doing back then. Then I did DNA profiling and eliminated over 500 people using that single locus test, which was the 3rd one in. Developed the PCR test that was the 4th one, and in combination between the 2, looked 7 cases together. Did a mass screen of 5,000 people and identified someone that way. And then over the course of the next 2 decades, effectively, we also did Y chromosome analysis, which you're going to come back to, biologic ancestry analysis as well. I've given evidence in this case 5 times, 5 separate court cases. I could draw you the crime scene right now on a piece of paper if you asked me to. But it was this case and the case that Sue Vintner had worked on, Operation Part, which led or really gave support to the formation of the DNA database, because clearly screening 5,000 people all the time is really not feasible. Can't really do it. So, the DNA database the legislation was established in 95 but enacted in 96. Malcolm Rawer, who was responsible for Susan Burdett's murder, was the first person to have a sample taken under the legislation. To get to this point, we had moved from 3 DNA loci to 6. Since then, we have gone to 10 to 15, 23 in global filer for some samples, 27 for YSTR. So we've added loci along the way since then. Each of that has added discrimination, specificity, and sensitivity, and almost all of it involves students from the forensic science program doing their master's projects in research for those developments. We loaded our first crime profiles in 1998, June the 30th, I know that date well, and originally our hit rate was 25%. So that means for every 4 samples we loaded, 1 linked. Since that time it's risen to 71, so for every 10, 7 link, and it's been like that now for a long time, a very long time. We also now have a Y chromosome crime sample database as well, which is somewhere around 1,000 profiles as of today, and that also has a link rate, although lower. So I just put 2 examples up here. The top example was not my case, belonged to my colleague Sue Trizet. That was a case that people know, the Teresa Cormack case, which was solved using the database. And the other one is Murray Jameson, and actually the case manager of that is right here as well. And that was the first time that we used familial searching with a successful outcome as well on the DNA database. So I put that one in as well as examples. And all this you can find in the Herald if you want to go and look at it in more detail. Okay, and the last thing that happened at this time, the forensic science program was established. And I started my journey back at Auckland University as an honorary lecturer in the program, which has obviously led me here. And I've got 2 of my lovely students on here, one of whom is in the audience, Catherine, somewhere, can't remember, up there at the back. Catherine and Tony Larkin were my first master students ever, and they were so awesome, I just took on so many more. Catherine works at ESR as a science leader in the STARMIX team, and Tony is in a really senior role in the UK in forensic science too. He couldn't be here, he did try, it was all too hard this time of year. So thank you, Catherine and Tony, for all that you did in that first year, because if it had gone to custard, I don't think I'd be standing here right now. So let's move on. So this part of the race is about consolidation. The harbour bridges, which is that picture there, obviously is 15 kilometres in, more or less. I just put up a snapshot of some of the things that happened. I'm going to talk about the first one and the last one in slightly more detail now. And the other thing that happened, which was pretty significant, was my 3rd baby arrived, Sebastian, who's not here. We couldn't choose between the photos. Eloise likes the top one, Ruben likes the bottom one. The elephant at Auckland Zoo has been a massive part of our lives. We have photographs from every time we have been there, and it doesn't matter how old they are, they always sit on the elephant, right? Right. Yeah, I just picked that one because he was a baby, and he was big. Okay. Of all the things I've done in forensic science, I'm going to say that the work we did to establish Y chromosome testing at ESR was probably the most impactful work that I have done. It started again with student projects in the early 2000s, and we started to do casework in 2004. We now use this testing in about half of all of our casework. It is particularly important in sexual assault cases, particularly those involving children, or complainants who come in a bit late after I've left it too late to come in. I'm sure you all know that your Y chromosome is passed from father to son. What you may not know is that although it has few genes, it has a lot of repetitive elements that vary a lot between unrelated males. And that's what we capitalise on in forensic science. It's really good for mixtures of DNA from males, and it's really good at family relationships and biogeographical ancestry predictions as well. So we did a whole heap of validation in that time. I've only got enough room for a couple of slides. This is the sort of stuff we do in validation. I know to some of you it sounds a bit maybe not terribly interesting, but it is absolutely crucial to underpin the services that we're providing in the core room. What we do has to be reliable. It has to be. We can't be going into court with stuff that's not reliable. And so a lot of our time in PHF science is spent on validation. A lot of our time. Sorry, John. Also, just for interest, really more than anything else, the types of samples that we get. So, and this is an old slide, but it hasn't really changed much. By and large, the majority are genital swabs from children and people for whom the time delay has been a bit long, or where there were no spermatozoa found. So that's the kind of stuff that we're working on. And I mentioned biogeographical ancestry. So this is a really old slide. Yeah, it's a really old slide. It's not the greatest. But anyway, why does this work? Well, this works, and you don't really need to know to read the legend here. All you really need to know is that some of the DNA loci on the Y chromosome, the populations in Oceania, so the Polynesians, the Pacific Islanders, the Cook Island Maori and Maori, are quite different at different sites to European and Asian populations. And this is just two examples from two different sites, and you can see that there are, in some places there's a nice big strong green peak, and in some places there's a nice big strong red peak. And it's those differences between populations that we exploit when we're doing something like biogeographical ancestry. And it's also why it's absolutely crucial that we have our own population database here in Aotearoa, because we have to model our own population. We can't use anybody else. So, if you ask my colleagues overseas, what am I most famous for? They would say body fluid identification, I think. My first foray into this came in 1992 with my first ever forensic paper, which was a protein paper, where I was looking at lactate dehydrogenase isoenzymes to distinguish between vaginal injury and menstrual blood. That was my first paper, and that was followed by a moment of absolute brilliance, which is why the light bulbs are there, from Douglas, who suggested I look at RNA as an alternative to the tests that we were using at the time. And so one of my students, Kim Tinker, came and did our first mRNA study and was very successful at distinguishing between venous blood and menstrual blood with the assay that she developed. So we're going to just go back a bit and say, well, why are we doing this? Why is this important? What is the problem we're trying to solve? So at the moment, with the exception of the RNA testing, which we do in a small number of cases, almost all of the body fluid kind of ID we do uses chemical tests, lateral flow tests. Unless we see sperm, which is obviously confirmatory, they're not 100% specific. They're very sensitive, but they are not 100% specific, and they do have a false positive rate. And so we report those with words like probable, possible, and likely, which is not terribly scientific. So that's one problem we're trying to solve. And the other thing is, well, why do we care? So you're probably thinking crime scene, blood everywhere. That's fine if that's the case, but often it's, in fact, most often it's not. The samples we receive are not obviously anything, and we need to know what they are in order to support the investigation. So that's why we care. So the solution at that time was mRNA. I'm not sure how much of this I need to go, but we're going back to my roots a little bit here to work with RNA again, because I hadn't done that since I left my postdoc. Viral RNA is going to become more important in a couple of slides' time, but this is just mRNA. So again, for the chemists, your DNA is the same in all of your cells, but your different cell types have a different RNA profile, and in turn a different protein profile. And it is a different RNA profile in people's biological fluids that we were wanting to exploit in this particular work. And mRNA had been thought to be unstable, because it is a very stable molecule. If you remember my reference to cold rooms, that's not true in a dried forensic stain. It's perfectly fine. It's quite happy. So my first paper is with my colleague Rachel Fleming, who unfortunately couldn't be here. And we developed the first iteration of this test, which was a multiplex PCR assay for blood, semen, saliva, vaginal fluid, menstrual blood. Yes, fine. And we did all the validation that you would expect us to do, so I don't really need to go through any of that, but pretty much the same as we did with Y, but this time with RNA. Since that time, other people have taken some of this on. So cell type 1, which is what we called it first, became cell type 2, and that was led by a PhD student, Patricia, working with Rachel. Cell type 3 is in development with Anna, who was here somewhere in the audience up there. But also, crucially, Anna, who was doing a master's project back then, came up with a method for co-extraction of DNA and RNA from one biological sample without destroying either, which nobody else had done before. So that was a master's student. Very proud of my master's student, Anna. And that was a really big development for the community, actually. And it's one of our highest cited papers, too. Okay, moving on. Marcus moved on. So, at 30K, you can hit the wall, or you can keep going. And in 2013, I had a PhD student called Julia Allwood, we published a paper on using SNPs, so single nucleotide polymorphisms, and classification trees, predicting hair and eye colour in a New Zealand population, which was the first time that was done. But that led to a more ambitious project, parts of which I'm going to talk about in a moment. And it started our sequencing journey in the forensic biology team in earnest. And our little project called Genomics at Mask was born. And our team site, we're big on teams at PHF Science, has 23 channels currently, representing all the work that we have done over the years in this project and are still doing. So I am going to talk very, very briefly with one sentence on each of these students. This is my dream team of PhD students. 3 of them are in the room. One of them's on holiday. So we start with Bethany. Bethany did her PhD on whole genome mitochondrial DNA sequencing for forensic applications with myself and James Curran, who's here. Then we have my wizard Alex, who is a wizard at bioinformatics and did a lot of development about how to recognise those short-term repeat sequences in DNA sequence. And he did that with myself and David Welsh on computer science. And then Rebecca, who looked at DNA methylation to distinguish between twins and to establish biological age. And then finally Brian, who is not here, who I'm going to talk a bit more about his project on the next slide. His project has led into another project, which is now quite important. So Ryan looked at this particular set of markers, so STRs, 58 STRs, so we talked about 10, 15, 20, 27 before, now we're talking 58, with some of the SNPs for identity, some ancestry and some phenotype markers. And you study a big population, so over 500 people, a lot of whom were volunteers from the University of Auckland actually, because him and Rebecca set up a stand in the bottom of 302. Oh, somebody's nodding at me. I'm not looking, more people are nodding at me. Okay. I don't know this, I'm not allowed to know. And yeah, so you're in that study, so if you want your answers, if you want your results, let me know. So on the left we have a SDR profile, we have a SNP in the marker, SNP in the middle. But I want to point out the stuff on the right hand side. So at the top we have 0.78 gram for hair colour and 0.95 for blue eyes. And then underneath is a probability, more or less. That's the easiest way to describe it. And then at the bottom you can see there's a red dot right in the middle of the purple cluster, and the purple cluster is the Europeans. So we have a brown-haired, blue-eyed European. Anybody want to guess? Not going to ask you with me. You can't really use other people's stuff right in the lecture. So there we go. But this is really important because the next thing that we've done is forensic genetic genealogy. And the people who are working on this at PHF Science, don't worry, I'm not going far. This is the use of direct-to-consumer databases in conjunction with DNA profiles from crime scene samples to identify the relatives of, and eventually the source of that profile, and using both the public databases and genealogy. I'm going to talk you just a couple of slides through this. So there are lots of challenges with this, not only technical ones, but also ethical ones, social ones, data governance ones, etc., which we'll touch on in a moment. I just wanted to briefly explain, and I cannot do this as well as Bruce did it this morning in our team meeting, but never mind. The basis of all of this is how much DNA do you share. So you'll know you inherit your DNA from your mum and your dad, which means you share DNA with your mum and dad. You also share it with your siblings, you share it with your children, and as you go out in your relationships, you share less and less and less. And we measure that degree of relatedness using something called centimorgans. So the more centimorgans you share, the closer the relationship is. So that's basically the guts of it. So what does forensic genetic genealogy really mean? And this is supposed to be a reassuring slide. We have to know that the sample probably did actually come from a suspect, not just somebody who happened to be passing by. So we have to have reasonable cause to know that this is something relevant to the case. We have to have done everything else that we possibly can before we go down this route. Currently at ESR we have implemented a method that sequences over 10,000 SNPs in the human genome in every sample. And I highlighted the ones in purple because they are the same ones that Ryan has done in his PhD. So that gives us some concordance data and some confidence, should we need it. And also a whole load of kinship SNPs. And this is a forensic specific test. It's been designed as best as it can be to avoid anything to do with health informative markers. Although if you're a health person, you probably realise that if you're doing 10,000 SNPs, there's probably one or two in there that may well turn out to be that. But just at the moment, not as far as anybody knows. So you get your profile, you upload it to one of these databases. GEBmatch is the one that me most use. And that comes back with a list of matches. And these are not matches from the GEBmatch database. These are actually matches from Ancestry, nothing to do with law enforcement. This is to do with me. But it just shows you the types of relationships that you will get. So a second cousin, this particular second cousin of mine, shares about 240 something centimetres to me. The next lady down, she shares 202. I know who she is. She is that relationship to me. I've known her pretty much my whole life. I didn't know she'd done Ancestry when I did this, but I do now. She reached out to me after we've done that. And then the genealogists take over and they build family trees and they work everything out that way. So that's not what we do. We just do the first bit. So obviously in Aotearoa there are some ethical, social, cultural things we need to take into account. Well, we would have anyway. And we did, and I was really proud of this work because I'd never done social science really before, but my colleague at ESR, PhD Science, Susan Manning, did this work, or led this work with herself and Anna mostly. And this was about workshops, interviews, all sorts of engagement with a whole range of people across academia, across the justice sector, across victim support, Maori academics, lawyers, ethicists, all sorts of people, to try and do some piece of work around what were the social and cultural. We all knew there were issues, right? We just needed something a bit more evidence-based in order to be able to sort of, well, design. So what we used this for was designing the processes and the procedures that we currently have in terms of how are we going to use the data, how are we going to store the data, what are the limitations and all that stuff that goes around that. So this is a really important piece of work, and I personally think scientists should be doing more of this, generally. It's not my area of expertise, but it was great to be involved in it. All right, sorry James and Bruce and Martin and others. There isn't much statistics in this talk, I'm going to say, and at grave danger of oversimplifying, but the interpretation of forensic evidence is based on Bayes' theorem. Some of you will know Bayes' theorem, and we deal in conditional probabilities most of the time. And we're working out what is the probability of my DNA evidence if someone is the source of the sample compared to if he or she is not. And I've been working in this space now pretty much my entire career, and have now taken this on to a new direction, expanded my direction. Here are the biggest questions in forensic biology today. What does the body fluid sample originate from? How did it get there, and when did it get there? So one solution to this is, gosh, I am a bit slow, aren't I? Source is reporting, how we report, evaluative reporting. Evaluative just means a likelihood ratio approach, I'm going to say. On the left, we have what we call a hierarchy of propositions. So we start at the bottom with a DNA mixture component, and then we have DNA, which is called subsource, the source of the DNA, blood, how it was left kicking, and the offence, which is nothing to do with us, which is to do with the cause problem. And we started our journey with source level reporting, because remember I mentioned that we just say probable, possible, likely. We wanted to turn this into something a bit more evidence-based with some data behind it. So you don't need to worry about the Bayesian network that you can see in front of you. But what we have done is we have transformed all of the data that we have available to us from the literature and work we've done at this ourselves, and put it behind these Bayesian networks to turn, instead of having a probable, possible, we have a likelihood ratio now. And the likelihood ratio is important because we turn it into what's called a global equivalent, and we do this for all evidence types. So now we can be consistent with the reporting of other evidence types, including DNA, but we can also incorporate other types of evidence types into the same structures. So the people that have mostly worked on this, but not exclusively, are listed at the bottom. Okay, back to the future. I could not do this talk without this slide. So, in last part of 2020, early 2021, myself, Rachel Ball, and Olivia Yukovic were asked to set up ESR was asked to set up a COVID sequencing lab at Mount Albert, and that's what we did. The three of us, that's what we did. We spent a few months on that, got it up and organised and running, and Olivia and I did the first case in January of 2021. And the reason for this was to do the samples from Auckland in Auckland because it was quicker, rather than transporting them to Wellington where the work had been done before. So our colleagues in Wellington got a bit of a reprieve because obviously we then had the Auckland outbreak, so we were doing all the Auckland samples and they were doing the rest of the country, and there were more in Auckland than we were down there. So we ended up doing the lion's share of the work through 2021. We expanded the team up to 6 with Ryan, who you've heard about before, Denisha and Claire. And to Ryan and Denisha in particular, thank you for sharing. You were my lovely family, thank you for sharing. Because we spent a lot of time together, locked in a room. And that is me, with a pipette, doing COVID sequencing. And I also wanted to shout out to Nikki Freed. I didn't see her before, I don't think she's here. Nikki developed the method that we used, and it went global. It was sensational and global and worldwide. And she worked still at Auckland University. She was at Massey at the time, and it was insane. It was the best collegial global effort that I've ever been part of. And I was very proud to be part of it too. Okay, nearly there. New beginnings. So I came here in 3 years ago, pretty much just over 3 years ago, to run the forensic science program. Since I started, we've done two things really that are different. We started to really give emphasis to Maori cultural values in the program, make them more explicit. I don't know that the students necessarily have noticed, but I have been trying my best to involve all of the principles that go with being Maori into the program, because it makes perfect sense in forensic science. And we've also introduced a new data and digital paper, recognising the change in forensic science globally into a data and digital driven era. And that paper is mostly taught by colleagues Janet, Zia, Ryan, who's not, and Jason. So there's no such thing as a finish line. And what do all good hobbits do after a long journey? They plan the next one, and if you run a race in that case, for me that was the Huffling Marathon, Hobbiton, where you run with other Tolkien nerds. Seriously, you visit the pub as you run, you get to meander through your happy place, but it's not for the faint-hearted. So it sounds like a good idea. Make sure you've done some training because it's hard. Yeah, it's hard. everybody's time is 30% longer than it would normally be. But it doesn't matter because everybody's happy. But what about forensic science? So we're nearly there, don't worry. So, my research manager's in the audience. I'm not sure what she's going to say to this. But I've turned myself back into a protein person, and we've started a project on proteomics. So the deal here is that some forensic samples are very difficult to deal with. I'm going to talk about 2 of them in a moment. But it turns out there are mutations, there are differences in your proteins just like there are in your DNA. You can do your proteins with mass spec, you can do your DNA with normal DNA stuff, and you can compare the 2. So if you've got a DNA sample from a suspect and a protein sample from a thing, you can compare them. Right? So that's what we're doing. That is what we're doing. So Sam and Orla come into this. So these are current master's students doing this exciting new stuff. Sam and Orla Sam is working on fired cartridge cases. Cartridge cases, big evidence type for ESR, you can imagine. The DNA doesn't like it being fired out of a gun. It's hot and metal and unpleasant. But the finger marks don't like it either in terms of the ridge patterns and things, but there's still protein there. So the idea is that we might be able to actually get some forensic evidence on these fired cartridge cases left at crime scenes and link them back to a person, potentially. So that's one project. And the other one is around hair evidence, very underrated evidence type. You can do it with mitochondrial DNA, but that's really expensive and time-consuming, sorry, Bethany. But you can do it with proteins as well. So that's another angle. I'm just going to skip that one because they don't have time, which is a shame. And that one. Okay. I think we're kind of out of time. Yeah, we are. So, colleagues in forensic science, all of you. It doesn't matter if you're a researcher or a caseworker or a vendor or what country you're in, the work that you're doing is impacting people and their communities every single day, and you should be very proud of yourselves. A lot of my friends are going to be watching the link because obviously they're not here. You should all be very proud of yourselves, whatever your role is. Younger people, look over there, because they're mostly over there. You can do this too. Find your tribe and you work as a team. That's what you do. That's my team up there. Sometimes you have to be patient and you have to time your run. When you fall down, you get back up and you walk on. That's a football reference. And be a hobbit, not an orc. Just be a hobbit. That's my bad end down the corner. So, to my family, one of whom is here. My cousins, Emily at the top, my kids' cousins and my kids in the middle, my brother on the other side, and the Harbisons collectively in the middle, including my parents. I'm not always there when I should be. But maybe now you can see why. Because for my children and my stepchildren, from the biggest to the smallest, the oldest to the youngest, my curious mind and my Disney princess, so proud of all of you. There's a lot. And to you that loves me best of all, Alini and Kalima, thank you for everything that you've done. And I'm going to leave you with this. I'm not going to read it out in Māori because you all know what it means. But to those of you who don't know what it means across the globe, what is the most important thing in the world? It is the people, the people, the people. And I think that is the most important message for a forensic scientist to know.